So I just want to welcome Rebecca Stewart. Rebecca's from Infection and Immunity, and she's going to talk to us today about Toxoplasma. And is my cat making me crazy?
Thanks for that. I'll just turn the lights down a little bit. Okay. So our lab, the Tonkin lab, works on this parasite called Toxoplasma. But you might have heard in the news a few headlines coming out about cats and this mind-controlling parasite. And so these news reports started from a couple of papers that have come out recently. So this one came out a few years ago, fatal attraction in rats infected with Toxoplasma gondii. And more recently, morbid attraction to leopard urine in Toxoplasma-infected chimps. And so these are the actual scientific papers which spurred a lot of these wild news article. Is your cat making you crazy? When love of cats turn toxic. But what's the science behind this? What does all of this actually mean? And so the life cycle of Toxoplasma, the sexual cycle does happen in cats. And so cats are an integral part of the life cycle of Toxoplasma. And throughout evolution, there's actually been a lot of feedback, or back and forth between cats who have the sexual cycle and rodents, so rats and mice, who possess the asexual cycle of the Toxoplasma. And I'll go through the asexual cycle in a minute. But humans can also be affected by Toxoplasma. And we most often pick up the infection from cats directly, so if we get scratched by them or we're changing the kitty litter, but we can also get it through meat. So if we're eating undercooked meat, the animal itself, the cattle, could have been infected with Toxoplasma and we're ingesting it if it's not cooked properly. And so then we get the infection, which is also the asexual cycle. So we know across the world there's 30 to 80% of human populations are infected with Toxoplasma, but quite often you won't know if you've had it. It's very mild or asymptomatic in healthy individuals. You might feel like you've got a bit of a cold, but most people don't actually know if they've had it. The exception to this is if you have any immune problems, you can have some clinical manifestations, and I'll talk about that a little bit later. But what about this Toxoplasma life cycle causes this sort of fatal attraction phenomenon that we're seeing? And it's this part of the life cycle that's really interesting in terms of this mind-controlling behaviour. Parasites differentiate into a slow-growing cyst form and remain with the organism for the remainder of its life. And they do this in neural tissue, so in our brain, and in muscle tissue. And so this is the asexual life cycle, also known as the lytic cycle. And these parasites are what's known as obligate intracellular parasites. So they have to get inside cells to survive. And so what they do is they find a new cell, so the host cell is us, or the cat, or the cattle. They invade, so they penetrate into the host cell, and once inside, they make themselves a nice little home called this parastophorous vacuole, a little comfy niche where they undergo asexual replication. This is what's known as clonal replication, because all of the daughter cells are identical in this case. And so once they're inside the cell and they've got a nice little home for themselves, this is where they differentiate off into the long-lived cysts in the neural tissue and the muscle tissue. And these are known as bradyzoites. So when we're looking at these parasites, they're tiny, they're quite small. So to give you an idea of the size, this is a human hair here. This is 10 microns, this little blue dot here. If we zoom in on that, so this is now 10 microns, that's what the parasite looks like. And so we do a lot of microscopy to do this. We zoom in to be able to see the parasites and assess what they're doing. And so here we have two parasites inside the host cell. So this is the host cell all the way around. And so you can see that after one having got in, it's replicated and now there's two in there. And so once they continue replicating, they create this really big parastophorous vacuole. So each of these is one little parasite. And again, they're inside the host. And this is a host cell nuclei here. So the nuclei is where all the DNA is stored, and this is where all the information for the cell happens. And this will be important a little bit later. Okay, so this work that I'm about to talk about was mostly done by a PhD student in our lab, Michael Coffey, and it's recently been published. And so we use this technique called fluorescent microscopy. So if you remember the big vacuole of parasites where there's lots of parasites in, here we can see them stained in red. So the outside of the parasites is stained in red. And remembering they're within the host cell. So you can see all of these parasite proteins within the host cell. And then here you can see the host cell in blue. So that's the nuclei of the host cell that I mentioned before. And so right now we're seeing the parasites are nicely contained in their vacuole. All the parasite proteins are within the parasite, and there's none going to the host. But if we have a look here, we have the parasites again, inside their vacuole, inside the host cell. We have some of the parasite proteins, but if we look here, that looks a lot like the host cell nuclei. So it looks like the parasite is exporting some of its proteins, some of its elements, into the host. And if we overlay the two of these with the host marker, we can see, in fact, some of these proteins are actually making it to the host nuclei. And so these are known as toxoplasma, like that toxoplasma exports these elements into the host cell. So what are these exported elements doing? Are they changing the host cell in some way? Are they modifying things in the host? And we know that once the parasites are inside the host cell, they like to make a comfortable niche for themselves. They don't want to be kicked out of their niche. So when they're sending these exported proteins into the host cell, they are changing a number of things. So this hypermigration of immune cells changes the immune system. This MHC class 2 downregulation, it means they can hide from the immune system. So the immune system isn't going to come along and boot them out. They're quite happy. They don't want that to happen. And this nutrient acquisition. So that's where they recruit energy for themselves for survival. So they sort of send out proteins that will bring back mitochondria or energy sources to them for their growth. So what does this have to do with this idea of behaviour modification? So in this paper here, they looked at this interplay between mice and cats. And what they found is that rodents infected with Toxoplasma, they exhibited behavioural changes. So there was a modified fear response. So they were more likely to venture out into open spaces. And they were actually would seek out cat urine. They're attracted to feline urine. So what does this mean in terms of the interplay between these animals? It means the parasites that are, the mice that are infected, they're going to be out in the open spaces. They're going to seek cats. And then the cats are going to eat them, and then the cycle continues. So there's this really interesting behavioural changes happening here. And then this has just recently been confirmed in chimpanzees and leopards. So it goes beyond just your domesticated cats and mice. It's all of the feline species. And considering chimpanzees are more closely related to us, is there an idea that this could be happening in humans or something along those lines? So in human health, there's actually some correlative studies seeing a higher incidence of toxoplasma in schizophrenics. And we see that people in risk-taking groups have a higher incidence of toxoplasma infection, such as when you do a retrospective study on car accident victims, you can see that that population is enriched for toxoplasma infection compared to the normal population. But it's really important to note that correlation, these correlative studies saying this happens at the same time as this, it's not causation. So as my boss has been quoted saying, no one's suggesting that toxoplasma is causing schizophrenia or bipolar, but it may be contributing to these diseases. And we've had some new studies come out recently that have found no correlation, so we're quite confused what's going on here. So the important thing for us to do now is understand the mechanism of this behavioural change. So here, what are these elements that are getting exported? What are they doing? How are they changing our behaviour? This is the mechanism of export. How are these elements getting out? Where are they going in the cell? What cells are they affecting? These are all the questions that we're asking. And so this work is ongoing, so we don't quite know the answers yet. This is where we are right now. And so this work is going to be continued by Michael Coffey, who I showed the picture of, and Simona, who's sitting down in the front here. So she's just started her PhD, so hopefully over the next couple of years we'll have some exciting answers to these questions. So just to switch tack a little bit, I did mention that in most people, infections are mild or asymptomatic, but there is a subset of people that will have problems with the acute infection of Toxoplasma, so that first instance of infection. And these are people that have a problem with their immune systems. And so what causes this is, we're going back to the lytic cycle, the parasites have found their host cell, they've gotten inside. This time, instead of differentiating into the slow-growing form, they actually keep growing, and they burst out of the cell, and they destroy the host cell in the process. And then this process starts all over again. It just keeps going round and round. And it's this process of exit from the host cell, what we call egress, that actually causes problems in the acute infection in people that don't have a very good immune system. And so here we can see in a brain. And this is a brain scan of an AIDS patient. So we know that when you have AIDS, your immune system is shot. There's not a whole lot of functioning immune cells left. And so we can see in the brain here, when it comes up again, we have a reactivation of this acute infection, which is causing this plaque in the brain. So we can see a lot of tissue damage, and that's from the parasites exiting, re-entering the cell, growing and exiting again. So this repeated round of tissue destruction. We can see that it actually affects pregnant women. And so if you're pregnant and you're infected for the first time, depending on the trimester you're in, there could be problems for your baby. So there's I think about 50 years ago especially, not so much anymore, a lot of pregnant women were warned not to change the kitty litter, that sort of thing. It's not really a huge problem in Australia, but it is something to sort of have in the back of your mind as well. And it also, in certain areas of the world, we have problems with ocular toxoplasmosis, so it'll get into your eye and cause problems with your vision. And this happens in South America mostly because of the certain types of toxoplasma they find there. They're a little bit more virulent. And so when we're looking at this, these clinical manifestations, as I mentioned, it's this part here, this exit from the host cell, this tissue damage that's causing the problems. And so we can actually look at this by live microscopy, and that's a lot of the work that I've done in the last 5 years that I've been in Chris Tonkin's lab. So again, we have, these are the big vacuoles of the parasites. So one parasite is probably about 32 here, probably about 32 to 64 there, and they're inside the host cell. And we can see, if we watch closely, you can see them start moving and they exit the host cell. So they burst out. And you can see some of them re-invading here, so we'll watch it again. So you can see here, this one here is re-invading the next host cell. You can see it pinching into the next host cell. So that's really nice. We can capture them. So we can capture them exiting. But we want to know a little bit more. What's the mechanism again? This word mechanism, that's what scientists love. What's the driving force behind this? And in this case, we know it's calcium. So if we remember back to year 12 chemistry, this one's calcium over here on the metal side. And we know in human health that it's important for bones. You know, when you're younger, you've got to drink your milk and eat your cheese. It's good for bone growth. But it's also really important in other biological processes. So it's important in the bone, it's important in muscles, it's important in neurons. And it often is important in this form of a calcium ion, so this calcium 2+ charge, because it commonly functions as a second messenger. So what do I mean by second messenger? Is that in a cell, often these calcium ions are sequestered. So they're stored inside a certain part of the cell away from everything else. And when there's a trigger, this calcium is released. And so it acts as a messenger to switch on things. So things go from this inactive or off state, calcium comes along, and it's now in an on state. So it's a messenger to say it's time to turn on, it's time to do something. So we also know Toxoplasma uses calcium, and it uses calcium in this egress part. And so now we've adapted a calcium sensor to detect this release of calcium in Toxoplasma. So again, what we have here is a vacuole inside a host cell. And so this calcium sensor is, I've got a heat scale here. So blue is low calcium, red is high calcium. And so what does this calcium profile look like during egress? So we can see the parasites activate their calcium before egressing. And you can see if you look closely, oops, you can actually see it's like asymmetric. It's not uniform, so it's quite interesting to see that. And so we can actually measure this, we can quantitate this, and we can see some graphs coming up like this. That's the calcium response. There's a nice peak. It's sort of held up high before the parasites egress. So one of my questions was, what happens if we take away this trigger or we block this trigger? What happens to the calcium response? Does it still happen? Are there other triggers? And so by blocking the parasite trigger, do we block calcium signaling? So here, I've treated these parasites with a drug that initially our hypothesis was that it's going to block the calcium signal. And so here we can see if it actually has. And so you see the vacuole here. It's a little bit of calcium signaling, but not much happening. Parasites don't really respond. There's none of that egress happening. And so we can see this video goes up to about 10 minutes, I think. Still nothing happening. And so when we quantitate that, we can see that this calcium response is nowhere near what we saw in the other cells. And when we do a comparison, you can see in the wild type cells, we see quite a robust calcium response. But in these cells where I've blocked the trigger, we don't see a calcium response. And we also don't see egress. And that's really important. If we're looking at treating these types of infections, we really need to block this egress. And so by blocking the calcium, we're blocking egress. Yes. Okay, so just to summarize what I've gone through today, a quick fly through of Toxoplasma. We know that cats are the definitive host of Toxoplasma. So humans, we can catch Toxoplasma from our cats or from ingesting undercooked meat. And generally, any infection for humans, if you're healthy, is asymptomatic. But now we're starting to see that Toxoplasma exports elements into the host cell. And so we're trying to understand what that is and what that means for human health and especially mental health, because these parasites exist in our brain. So we have some evidence which is correlative but not causative to show that there may be a link between Toxoplasma and human health. And we've still got more work to do on this. So maybe in a couple of years' time we can get Simona back and give you an update of what's going on. And then I briefly went through calcium as a second messenger in Toxoplasma and how we can block that and block this asexual cycle to stop any sort of manifestations of trauma or clinical events in the acute infection. And so this is Chris Tonkin, this is the lab head, so if you see him around, I think it's his birthday today actually. So Michael Coffey, as I mentioned, did the work in the first part of the talk. Alex and Melanie have been part of the lab for the last couple of years, and we've got a few new recruits now. These are our 4 new students, just started. And of course, none of this work could be possible without the imaging department. We've got a suite of amazing microscopes that are fundamental to any of the work that we're doing. So this is a huge part of the work we do, and these people really contribute to that. And so I'll leave it with that, and I'll take any questions if we have any. Yes?
Might be a dumb question, but does it do any physical damage, or does it actually make you like more mental, like does it have more of a mental illness?
So it does both in the different settings. So when we have that egressing, that causes tissue damage, and it can cause like the plaque in the brain, for example. So that's what we call the acute infection. So that's one side of it. And so it is causing tissue damage in that instance. But when it's gone into the cyst form, it's slow growing, it stays inside the host cell, it doesn't come out, and it sends out these export elements, and that's what we're thinking is changing the behaviour. So one of the things we're thinking is it might affect dopamine. So dopamine is one of these feel-good hormones, and if it's affecting this, does it change our behaviour? Is it sort of a reward for us? Is it telling us to do more risky things? That's one of the questions I think we're going to be looking at. So in that case, it's not the tissue damage, it's the things that they're sending out that are causing the problem.
Because it said fatal in the rats. Yes, so how did the rats die?
So there's different strains as well. So
Okay.
the ones that don't, we have one. strain that doesn't actually form these cysts at all. And so what happens is they die from the acute infection. So it's that sort of excessive cell damage, tissue damage from that exiting and reinvading. And so far all the work, or most of the work we've done in our lab has been using that acute strain that causes acute infection. So it's the tissue damage that's causing the problems in that one. And soon we'll be switching to the bradiozoites, so that's what Simone will be doing. And it will be looking more at sort of the behavioural changes, and so that's less tissue destruction and more of the exported elements. Yeah.
When you talked about the calcium and what you figured out to do with the drug to turn that calcium off, is there risk of turning other cells' calcium off and kind of destroying what those cells are supposed to do?
That's a great question. Because as I said, calcium's important for us too. So we don't want to affect our calcium because it's going to make us sick. So we really need to target these drugs carefully that will affect the parasite and not the host. And this is the same with all drug treatments. So if you think of chemotherapy, for example, generally we want to target the cancer and not us. But a lot of people do lose their hair, they do feel quite sick because there's those off-target effects that are getting to us. So when we do our drug design, we think carefully about what can we target in a parasite that won't affect a human. And so we keep that in mind. We're not quite there with these drugs yet. We still have a way to go, so we still need to do comparisons for that. And that's what clinical trials and things like that are for as well. So making sure it's not toxic to the host. So there are ways we can do that, yeah.
So when you say that it's asymptomatic if you've got it, so people wouldn't know that they have it?
Generally not. So what happens in our lab is before we start culturing them, they give us a blood test to see if we've got it. So everyone that comes through our lab gets a blood test. And what they're actually looking for in that blood test is antibodies to Toxoplasma, so it's to show that you've had a past infection. And so I know I mentioned that 30 to 80% of people have it. In our lab, no one so far has had it. So all of us that's been tested haven't had it. But yeah, you wouldn't know unless you're otherwise have immune problems. You generally wouldn't know. You might have a little bit of muscle soreness, but you'll probably put it off to, oh, I just got a bit of a cold and just a bit run down. You really generally don't know that you've got it.
And is it something that the person would clear, or they would have it for the rest of their life?
So you generally clear the acute infection. Your immune system's really good at clearing that acute infection. But it's when they switch to the bradiozoites, the slow growing, because they're hidden inside the host cell, and as I mentioned, they modulate the cells so they stay hidden, and so they sort of trick the immune system into saying, oh, I'm not here, just ignore me, I'm fine. It's that sort of slow growing that stays with you for the rest of your life. So we don't actually clear that side of it. And that's why, again, we can pick it up in meat as well, because those animals don't clear the infection, it will stay with them for the rest of their life. So.
Would most people who have cats have it?
Well, in Australia, it's actually not all cats have it as well. So only again depending on where you are, about 30% of cats might have it. And so you often find that cats that are predate, that like maybe they can roam around. Yeah, they roam. I don't have a cat, so I don't know the terminology, but or wild cats or feral cats, they're more likely to have it because they feed on the rodents and that sort of thing, so they're going to be exposed. Whereas your domestic cats, you know, they've got plenty of nice, clean food, so they don't really need to sort of seek out rodents as a source of food. So in Australia, the incidence is quite low. In countries like in South America, like Brazil, like I mentioned, it's a little bit higher. You do have more of these feral cats. You do have, you know, more of this sort of cycle happening. So yeah, not all cats have it. Not all people who have cats who have it will get it.
Yeah.
It's, you know, so it's sort of, your chances are higher of having it if you have a cat, but it's not to say that you absolutely have it if you have a cat.
So have there been any studies done on cat people versus dog people? And whether more cat people have toxoplasma?
Well, there actually hasn't, and so when you see a cartoon like this, you're sort of like, we should do that. Let's have a look and see what's going on. And I think that's sort of where the field is heading, because you know, we're starting to understand these sides of things more, and we're starting to, you know, realise that mental health problems are really important, whereas before they're sort of brushed aside a little bit. And so now that's where the research is going and that's where the push is going. Whereas 10 years ago, maybe it was on something else. So I feel like the next 5 to 10 years, we're going to have some really exciting science coming out on this side of things. We're going to have a few answers, hopefully, about what's going on. And we can sort of go from there in terms of therapeutics. So.
Is that why crazy cat ladies get loads of cats?
Yeah. I mean, that's sort of, you know, you see that and you sort of think, well, is there something going on there? It's a good question to ask.
Is it something you can get checked for?
Yeah, you can. Like, if you want to get tested, the staff at the clinic, that's where I got tested as well. So you can do that. It's just a blood test. So yeah, there's not like if you've had it, there's nothing really changes. You just don't. Okay, all right. I think we'll leave it there. Thanks for coming.